Hello, my name is Mydia, and in this video, I'll be talking a little bit more about interneurons and what their development process is like, especially with the different types of interneurons. To give you more information, interneurons are neurons that connect between two other types of neurons. They're only found in the central nervous system, and most of the time, they transfer signals between sensory neurons and motor neurons along the spine. In the brain, interneurons make up about 20% of the neurons and tend to function in local brain areas, working to create circuits in the neocortex. The development of interneurons starts initially with neurogenesis of the precursors in the ventricular zone, then goes through two different migration processes to get to their destination. Initially, a tangential migration from the ventricular zone to the neocortex, then a radial migration to the specific lamina in the neocortex before they go through the maturation process, which consists of programmed cell death in order to hone down to the right amount of interneurons and through circuit refinement, where they correct synapse formation and firing. Interneuron identity is likely specified at neuronal birth, but it unfolds over a period of time where the final characteristics of each type of interneuron are acquired as time goes on. Before we can talk about the emergence of interneurons, we start with the neuroepithelium during initial neural tube closure. Then, as the telencephalon forms, the neuroepithelium develops into the pallium and subpallium during E3. The subpallium is what develops into the four origin sites of interneuron progenitors, and then go on through the development of other prominent parts of the brain to form the interneuron types that we see at the top here. There are four neurogenic sites that include the three ganglionic eminences: the lateral ganglionic eminence, the medial ganglionic eminence, and the caudal ganglionic eminence. The fourth one is an additional area near the medial ganglionic eminence called the preoptic area, or the POA. As interneurons develop from the caudal and medial ganglionic areas, as well as the preoptic area, there are specific transcription factors influencing the expression and eventually what cell fate occurs for each interneuron. DLX1 and 2 are in all regions, but factors such as NKX21 or sonic hedgehog give rise to specific types of interneurons that originate from the same progenitor area. These transcription factors and their opposing gradients will then cause a second layer of genes to be expressed for each specific interneuron created. Out of the GABAergic interneuron neurogenesis sites, researchers now believe that about 70% of cortical interneurons come from the medial ganglionic eminence and the preoptic area, while the other 30% come from the lateral ganglionic eminence and the caudal ganglionic eminence. This picture shows the different types of cortical interneurons, which include the parvalbumin, the PV, somatostatin, SST, or the 5-TH3AR serotonin receptor expressing interneurons. And within each of these different types of interneurons that express different factors, there are specific interneurons within each category, some of which include chandelier cells as PV expressing, Martinotti cells that express SST, and basket cells that are in all three categories. This shows how the distinction between the different categories are much more fuzzy than the categorization of excitatory or inhibitory neurons. Interneuron diversity can be organized from a combination of different transcriptional modules during development, and researchers today organize the different types of interneurons through their morphological, biochemical, intrinsic, and connectivity properties. We can also see here that there's a slight preference for which progenitor regions create different types of interneurons in the neocortex, but it's not as distinct or clear-cut, and more studies are needed to be done in order to further categorize these interneurons.